Dr. Launen of Keene State College, and I'm going to continue this lecture on transcription, talking about elongation briefly, and then termination of transcription. So elongation means to actually build the RNA transcript, which means selecting the correct RNA nucleotides as shown here, and forming phosphodiester linkages in between each of the RNA nucleotides. And this process is governed by base pairing. So it's governed by DNA, RNA base pairing, using the rules that I discussed in earlier parts of this lecture. So the template is utilized to select the correct RNA nucleotide, and the formation of the covalent bonds called phosphodiester linkages between RNA linkages is catalyzed by the enzyme RNA polymerase. So RNA polymerase catalyzes the formation of phosphodiester linkages, and base pairing governs or controls the identity of the nucleotides. And that's really all there is to say about this, other than RNA polymerase is a pretty cool enzyme in that it is able to create and maintain a transcription bubble shown here, and also synthesize new RNA all at the same time. It does that by adding nucleotides to the 3' end, so the 5' end comes out of the transcription bubble first, and this whole structure will move down the template, traveling 3' to 5' down the template. Okay? All right, let's talk about termination now. So we're going to focus really just on termination of transcription in bacteria because scientists know quite a bit about that. We understand that process fairly well, and we understand the process of terminating transcription in eukaryotic systems much more poorly. So for our purposes, I'm going to stick to termination in bacteria, and I'm going to focus on two basic modes, intrinsic and what's called Rho mediated. That's how you pronounce this protein here. So intrinsic termination of transcription means you synthesize RNA that will base pair internally, thereby creating a hairpin structure, which causes RNA polymerase, Pol is short for polymerase, to dissociate or come off the gene. That's one mode. Okay? The second mode is mediated by proteins that are called Rho proteins. And so these are proteins that will bind at specific sequences on the 5' end of a gene, at the end of a gene, like after the stop codon and in the 5' UTR. And so that is another way in which termination of transcription can occur in bacteria. So let's take a look at that first mode, intrinsic termination. So here is our DNA gene, okay, and the gene is going to be at the very end of the gene. There's going to be spacer sequence. There's going to be what are called inverted repeats, and they're shown in yellow here. And you'll note that they're mirror images of one another, okay, from left to right. And there's a little spacer sequence just to create a little bit of room here. And then there's a poly A sequence, okay. And that's not actually seen in all bacterial systems. It's common in eukaryotic systems, but not so much all bacterial systems. So that's kind of an oversimplification here. So what will happen is when this sequence is utilized to make corresponding messenger RNA, which is shown here, spooling out of the transcription bubble, eventually enough of it will come out, and remember this is in the watery environment of the cell, and this RNA molecule is single-stranded, so it's kind of flopping around in there. And what will happen is this region and this region, which were produced when you transcribed this and this, which remember they were mirror images of each other, they are going to find each other just randomly, and they're going to be chemically attracted to one another, and they're going to form the hydrogen bonds known as base pair, which is going to stabilize this structure because those are quite a few hydrogen bonds in a row. And this creates a structure that's referred to as a hairpin loop. And there's something about that hairpin loop that actually disrupts RNA polymerase, which is like riding along here in the transcription bubble, and actually forces it off the gene, causes it to dissociate from the gene, which releases the RNA transcript, and the rest of it's going to be in here. It releases the RNA transcript and leaves it free to go off and be translated. Correction to what I said earlier, the rest of the RNA transcript's actually out here, not over here. This is the very end, okay? And that's how transcription is terminated using intrinsic sequences through the formation of hairpin loop structures. The other mode, just to go back for a minute, is to utilize Rho proteins, which bind to specific termination signals at the very end of the RNA transcript and the associated template, DNA template, causing RNA polymerase to dissociate. And I don't have an image to show you of that, but that's the text that goes with it. All right, so what we're going to do next is we're going to move on in the next part of this lecture to talk about RNA processing in eukaryotes.